In this video tutorial, we're going to demonstrate how to use the oil immersion bright field microscope. The bright field microscope creates an image by focusing a beam of light through a specimen, and then diffracting the beam with curved glass lenses in order to magnify or enlarge the image. Your microscope has an LED light source located in the base. The light is turned on using a switch on the side of the scope. There is also a dimmer knob allowing you to control the intensity of the light. You'll generally want to dim the light at lower magnifications while having it brightest at higher magnifications. The beam of light initially travels through a lens located underneath the stage. This lens, called the condenser, serves to focus the light through a small area of the specimen. The funneling of light through the specimen ensures a high-quality image. For most scenarios, you'll want to have the condenser as high as possible. The elevation of the condenser can be controlled with a knob located on the side of the scope. The amount of light passing through a specimen can also be controlled using the diaphragm. The opening of the diaphragm can be increased or decreased as necessary using the small lever attached to it. Samples adhered to glass slides are referred to as specimens. Specimens are placed on the stage and secured with the stage clip. If the specimen is not located directly over the light source, the stage can be moved left or right and forwards or backwards using the stage control knobs. It will be important to have the specimen directly over the light source, as the light traveling through the specimen will then pass through one of 4 objective lenses. The objective lenses magnify the image. They are connected to a revolving nose piece, which allows each lens to be moved into position. The shortest objective is called the scanning lens. It has a red line and magnifies an image 4 times. The image will then pass through the ocular lens before reaching your eye. The ocular lens always magnifies 10 times. The magnification effects of the objective and ocular lenses are compounded or multiplied. Thus, with the 4x scanning objective in place, the total magnification of the image will be 40 times. The next smallest lens is the 10x objective, sometimes called the low-power lens. It has a yellow line and when compounded with the ocular lens provides a total magnification of 100 times. The next lens is the 40x objective, also called the high-power lens. It has a blue line and when compounded with the ocular lens provides 400 times magnification. The final lens is the 100x objective, also called the oil immersion lens. As will be discussed later, immersion oil is required at the highest magnifications in order to minimize the scattering of light. This allows for maximum resolution of the image. It is important to remember that this is the only lens designed for use with oil. When used correctly, the oil immersion lens, compounded with the ocular lens, provides 1,000 times magnification. The objective lenses can be focused using the coarse and fine adjustment knobs. These knobs control the elevation of the stage in relation to the objectives. You will always begin focusing using the coarse adjustment and then fine-tune your focus using the fine adjustment. Okay, if you feel comfortable with the parts of the microscope, let's begin the process of viewing a specimen. We'll be using a bacterial smear for this exercise. You'll first want to ensure that the scanning objective is in place and that the stage is at its lowest level. You can lower this stage using the coarse adjustment knob. This is also how you will leave the microscope when you are finished using it. We always begin with the 4x scanning lens because it has the largest diameter of field and the greatest depth of focus. This makes it the easiest lens to focus. Secure your specimen on the stage using the stage clip. Now bring the stage all the way up using the coarse adjustment knob. Turn on the light source and adjust the dimmer to a comfortable intensity. Now begin bringing the specimen into focus by turning the coarse adjustment and slowly lowering the stage. Get the specimen focused as best you can. It may not look perfect, but you can fine-tune things next using the fine adjustment. There, that looks pretty good. Recall that with the scanning lens in place, you are looking at an image that is magnified 40 times. Large dark areas of stain are sometimes the easiest areas to focus on at these low levels of magnification. We're now ready to try the 10x objective lens. Your microscope is parfocal. This means that the focal lengths of the objective lenses have been calibrated to allow each of them to be in focus at the same time. Simply rotate the 10x lens into place, and you'll find that the specimen is already nearly in focus. At this point, you can simply use the fine adjustment knob to bring the specimen into clear focus. Got it. Although the large stained spot helped to focus the specimen under the scanning lens, it will present a problem as we continue to increase magnification. This is because the diameter of field will continue to get smaller, and we don't want the entire field to be filled with that spot. Let's move the slide over to better center the specimen using the stage control knob. We're now ready to move the 40x objective lens into position. Once again, the image should already be nearly focused, so we'll simply use the fine adjustment to bring it fully into focus. There. At 400 times magnification, we can begin to see the individual cells. It's now time to discuss the 100x oil immersion lens. As the diameter of field continues to get smaller, there is less area for light to travel through the specimen and create an image. As such, it becomes very important to maximize the amount of light passing into the objective lens. There are a few ways to accomplish this. Elevating the condenser helps focus as much light as possible through the specimen. We did that at the beginning. If you haven't already done so, this would also be a good time to adjust the dimmer switch to provide maximum light intensity. Nonetheless, there remains a certain amount of light scattering that occurs when the light passes through the airspace between the specimen and the objective lens. Placing a drop of oil into this space can minimize the amount of scattered light, thus providing an image with higher resolution. This is due to the higher viscosity of the oil as compared to the air. It's as if we have filled the space with a malleable form of glass. Let's give it a try. Rotate the nose piece of the microscope to a position in between the 40x and 100x objectives. This will give you some room to work with on the stage. Without removing the slide, place a small drop of immersion oil onto the specimen directly above the light source. Now, rotate the oil immersion lens into place, being sure to not let the 40x lens touch the oil. Once again, the parfocal nature of the microscope means that we will only need to work with the fine adjustment knob to bring it into focus. There, we did it. We're now viewing bacteria at 1,000 times magnification. Here we can clearly see the bacillus shape of this particular species. When you have finished viewing the specimen, it's time to clean up. Turn off the light and rotate the 4x scanning lens into position. Be very careful not to allow any of the other objectives to contact the oil. The oil immersion lens is specially sealed to prevent oil from entering it. The other lenses are not sealed and are easily damaged by the presence of oil. Lower the stage all the way down using the coarse adjustment knob and remove the specimen. Place some vinegar solution on a Chem wipe and gently clean off the ocular lens as well as the 4x, 10x, and 40x objectives. Use another Chem wipe with vinegar to clean off the oil immersion lens. You may want to wipe this lens with a dry area of the Chem wipe when finished to ensure that all of the oil has been removed. That's it. You're done. I hope you found this tutorial helpful. The fascinating world of microscopy now awaits you. Good luck!